Friends, welcome to Biology Made Easy. Today we are starting a new chapter, that is Principles of Inheritance and Variation. This chapter is coming under genetics, and this is a very interesting chapter. It is also very important from examination point of view. So I hope you will enjoy the entire chapter. But before entering into the chapter, today in this video, we will discuss some important terms, because unless you understand these terms, then understanding other things becomes very difficult. So let us start with some important terms so that your understanding will be very clear. Now, first thing is we will discuss here principles of inheritance and variation. So first we have to understand what is inheritance and what is variation. So the first point that we will discuss is inheritance. You know one proverb, like begets like. That means a particular kind of organism, maybe plant or animal, produces its own type. When we put one mango seed in the soil, it gives rise to a mango plant. One elephant gives rise to a baby elephant. One human being gives birth only to human being. That is like begets like. How it is possible? It is possible because of transmission of characters from parents to offsprings, generation after generation. This is known as inheritance. That means transmission of characters. Transmission of characters from parents to offsprings, from generation to generation, or we can tell in successive generations, from generation to generation. This is known as inheritance. And heredity, the heredity is nothing, it is only inheritance. That means we can call that inheritance is the basis of heredity. Inheritance is the basis of heredity. Now the second term is variation. And we are all acquainted with this term variation. You see, in a family, there are 3 or 4 brothers and sisters are there. But there is a lot of variation between the brothers and sisters among themselves as well as from their parents. The parents are same, but why such a lot of variations are there? It is because of sexual reproduction. Because during sexual reproduction, the crossing over or recombination takes place, which is responsible for this. And the earlier the agriculturalists, there are those who are living around 8,000 to 10,000 BC, they are also aware of these variations, and according to them, that the sexual reproduction is responsible variation. That is why they were breeding some of the domesticated plants and animals, and then they are selecting. By this method, the present day chicken has been obtained from wild fowls as a result of selective breeding. Similarly, the Filial cow, which is very famous in case of Punjab, it is also obtained from wild cows. So it is that because of the domestication and breeding and finally artificial selection. So these variations usually arise due to recombination or crossing over. It may be due to mutation. Also, it may be due to environmental reasons. So now 2 things are clear. What is inheritance and the variation? Now the third term is genetics. What is genetics? Genetics is a branch of science which deals with heredity and variation. That means genetics is a branch of science which deals with the heredity and variation. And what is heredity? It is inheritance. That means the transmission of characters from parents to offsprings, and variation we have discussed. So when these two are combining, that is genetics. Now the fourth term that we will discuss is the factor. The term factor was coined by Mendel himself, because he is the father of genetics. So factor. So what is this factor? According to Mendel, the factor is the smallest unit which carries the character or trait from one generation to another generation. That means it is the smallest unit of inheritance which passes from generation to generation. Now the next term is gene, and the term gene was coined by Johansen. What is gene? The term factor that was used by Mendel is nothing. That is the presented gene. That is coined by Johansen. So what is a gene? Again, just like this, it is the smallest hereditary unit. It is a segment of DNA. And nowadays we are calling it as cistern. It is a segment of a DNA having a particular function. Next, it is a polynucleotide chain. Because when it is a part of a DNA, so naturally it will be a polynucleotide chain. It is present on the chromosome at a specific position called a locus. Present on chromosome at a specific position, and that specific position is known as locus. Locus is the point where the gene lies. The next important point is it contains information, has information for translation. That means what type of proteins and enzymes will be synthesized, that information is contained by this gene. Then the next point. Usually the genes are designated by one or few letters. Suppose we will write T. So it represents the height of the organism. When we are writing around R, we are writing R, then it is representing the shape of the seed. That means single letters are there. But sometimes you use the word cry. Here 3 letters are there. So this also represents one gene, and this produces the cry gene produces cry protein. So it is usually represented by one or few letters. Then let us discuss the next point. So the next point that we will discuss is usually allele or allelomorph. So allele or allelomorph, the term also coined by Bateson. And what is the meaning of this allele or allelomorph? The meaning is one another. That means a gene has different variants. It can give rise to one character, but when it is modified, it can give rise to another character. That means, suppose capital T, it will give rise to the tallness. But when small t is present, it will give rise to dwarfness. That means both capital T and small t are two alleles of the same gene, that is the height of the organism. This is known as allele or allelomorph. That means two variants. When capital T is present, it represents tallness. When small t is present, it is represented dwarfness. And both tallness and dwarfness are two alternatives of the same gene, that is the height of the organism. So this is known as allele or allelomorph, and usually both of them are present in the same locus. Suppose in one chromosome here capital T is there, then in the homologous chromosome in the same position small t are present. That is, both the allele are present in the same locus of homologous chromosomes. That is important. Then the next point that we'll discuss is dominant and recessive. So the next point now we'll discuss is the dominant and recessive. Dominant and recessive. Now what is dominant? Dominant means when a character is expressed when it is present either in homozygous condition or in heterozygous condition, that is dominant. That means a character is both. Suppose let us take the gene T for height of the organism. If it is present in this manner homozygous, or it is present in this condition. So in this case also it represents tallness, in this case also it represents tallness. That means the homozygous condition it represents the character, and the same character is also represented by the heterozygous condition. This is known as dominant. But what is recessive? When the character is expressed only when it is present in recessive condition, not in a heterozygous condition. So when it is present in this, then it represents dwarfness. So when a character is expressed both in homozygous and heterozygous condition, it is dominant. But when it is represented only in the homozygous condition, it is recessive. This is one type of difference. There is a second type difference. In a heterozygous condition, the one which dominates the other one, which masks the expression of other one, that is dominant, and the one which is suppressed is non-recessive. So in both ways you can express the dominant and recessive characters. Now, the next point that we will discuss is the genotype and phenotype. Genotype and phenotype. Now, what is genotype? Genotype is genetic makeup. This is genetic makeup of an organism. So, genetic makeup of an organism, that means suppose we will write capital T. It is homozygous. It is homozygous tall. If we write capital T small t, it is heterozygous tall. If we write small t, it is homozygous dwarf. That means from this nature, this is the genetic makeup. From this genetic makeup, we can tell what is the type of the organism. Because from this, we are telling that it is homozygous tall or it is heterozygous tall or it is homozygous dwarf. This is the genotype. But what is phenotype? Phenotype is the observable character. Observable character. That means which you can see by your eyes. That is, suppose the height of the organism, the color of the body, the shape of the fruits or seeds, or we can see in by our eyes. That is observable characters. So that is known as phenotype. This is the basic difference. But again, one important thing is That is the phenotype. What will be the phenotype of the organism? That depends upon the genotype. So phenotype is equal to genotype plus environment, because environment has also influence, plus the interaction between genotype and environment. That means what will be the phenotype? That depends upon these three things: the genotype, the environment, and the genotype environment interactions. So now I hope now your concept is clear regarding the genotype and the phenotype. Then the next point that we will discuss is the homozygous and heterozygous. Homozygous and heterozygous. So these terms also coined by Bateson. Now what is homozygous and heterozygous? Now the genotypes, we are just to discuss genotypes. The genotypes can be homozygous or heterozygous. It may be homozygous or it may be heterozygous. So when both the alleles are similar, like this, capital T or small t, then we will call it homozygous. And when they are dissimilar, one capital T, small t, this is heterozygous. That means the genotypes are of two types, maybe homozygous or heterozygous. But sometimes a third term is there, that is known as hemizygous. What is hemizygous? When one in a diploid organism, only a single allele is present. So single allele in a diploid organism. So where it is possible? So it is possible in case of the X-linked genes, because in case of the male mammals, it is X and Y, the sex chromosome. Suppose in X some genes are there, that means only one there, because the other one is Y. So they are known as also hemizygous. Then some other terms are there. Then the next is filial. What is the meaning of a filial? Filial means offspring, or you can call son or daughter. That means when two parents produce some offsprings, they may be male or female, they are not, that is known as filial. So usually we write F1, that is first filial. First filial means first generation. When we write F2, that means second filial, that means the second generation. Like that, the filial term is there. Then the next point that we will discuss is the pure line. This is also very important, because when Mendel conducted his experiment, he took some true breeding or pure line ones. What is the meaning of this? Suppose we take one plant like this, genotype is like this, and another plant will take like this. So this is also tall, this is also tall. But we cannot know whether it is homozygous tall or heterozygous tall. And we want that the plant should carry only the tall character. Now see, if we take this plant, there will be no problem, because this is homozygous tall. But if we take this plant, because we cannot see what is the genetic makeup, so in the next generation, again it will give rise to tall and dwarf plants. Then again this may give rise to tall and dwarf plants. That means it is heterozygous. So if we are taking a heterozygous plant, how will we get pure lines? That means we have to self-feed for several generations. Till 4 to 6 generations we have to self-feed till we get our desired character. If we want only tall character, that means only when the tall plants will arise, dwarf plants will not arise, it is confirmed that it is now it has become pure line, it has become homozygous. Because in all breeding experiments, the homozygous characters are taken. Otherwise, we cannot get our desirable characters. Then another point is Punnet square. So Punnet square is just a checkerboard. A checkerboard just like one chessboard, and it is designed by the British geneticist Punnet. British geneticist Punnet has coined the terms. So, what is the importance of this? It is just like a checkerboard, and this checkerboard usually in this line, horizontal line, the male gametes are written, and in the vertical line, the female gametes are written. So, what is the probability of different offsprings? That can be determined from this. What is the probability? That is the importance of this Punnet square. So, these are some of the important terms that we have discussed in this video. And from this also, we can discuss some MCQs. So, one MCQs. Number one, we can write a character which is expressed in a hybrid. A character which is expressed in a hybrid is dominant. Second choice, codominant. Third choice, recessive. And the fourth choice, epistasis. This is one question. Let us discuss one second one. Alleles are first choice, different phenotypes. The second choice, true breeding homozygotes. Third choice, different molecular forms of a gene. And fourth choice, heterozygotes. And this question was asked in AIPMT 2015. You will be able to answer the questions because we have discussed all these things. So thank you very much for watching the video. God bless you. Have a good day.